Hi, thank you so much, organizers, for having me here. I will be talking about delivery of extraterrestrial ribose by meteorites. Jamie did a nice summary of amino acids in meteorites. Other than the amino acids, many organic compounds, including nucleobases, have been detected from meteorites. Sugars are also key molecules for life. Sugars support many biological reactions. We can see many sugars in biological molecules such as RNA, DNA, ATP, and many other functional molecules. So ribose is especially important as the sole sugar of RNA, but the delivery of ribose to the Earth has been unclear. So when we look at the synthesis on Earth, many investigations indicate the synthesis of ribose and other sugars by Formose type reaction on prebiotic Earth, although its accumulation remains unclear. So unlike amino acids and nucleobases, the source of ribose has been limited on terrestrial synthesis. But formaldehyde is a primary source material of sugars. This molecule has been detected from interstellar region by telescope observations. Formaldehyde was also detected from meteorites in the cometary coma by laboratory and in situ analysis. The glycolaldehyde is hydroxyaldehyde, but not a sugar, a little more sugar-like aldehyde than formaldehyde. This molecule was also detected in space by telescope and spacecraft observations. So source material of ribose and other biological sugars seem to be abundant in space. And laboratory experiments further support the synthesis of sugar in space. The previous research did UV radiation on interstellar cometary ice analogs and found the formation of many sugars, including ribose, deoxyribose, and others. So ribose might have been a ubiquitous molecule in space. But this has not been clear. Many sugar acids and sugar alcohols have been detected from meteorites, including ribitol and ribonic acid. These molecules have quite similar structures and compositions to ribose, but the sole extraterrestrial sugar detected from meteorites was this dihydroxyacetone, which is 3-carbon sugar and completely different from ribose and the other pentoses 5-carbon sugars. While the investigation of sugar in meteorites have been done for a long time, there has been a technical difficulty in sugar analysis. In general, meteorites contain a huge variations of organic compounds, many amino acids and other many organic compounds. And sugar is one of them, but sugar have many isomers. For example, these 4 ribose, xylose, ribinose, decussose, they have different sugars, but they are isomers having the same compositions. These when we look at just ribose, there are 5 configurations: beta ribopyranose, alpha ribopyranose, beta ribopyranose, alpha ribopyranose, and aldehyde form. For the analysis of sugars by gas chromatography, we needed to treat these sugars with chemicals to convert them into volatile compounds. This conversion is called derivatization. And if we fix one sugar molecule into these many configurations. By derivatization, many forms, many signals appears in the analysis. For example, even just 4 sugar potentially makes 16 isomers. And thus, lots of peaks signals appears in chromatography separation like this. So this analysis, and it's difficult to distinguish different sugars by chromatography. Here in this paper, by Maynard et al., they did this traditional derivatization, but avoid this difficulty by using extremely high resolution two-dimensional gas chromatography for separation and found ribose in their synthetic products. We did a different approach. We derivatized sugars to form one derivative from one sugar. We fixed all these configurations into linear form by the derivatization, which is different from the traditional derivatization. And by this derivatization, we can get these clear, very simple separation of chromatogram. And this separation also have advantage. We got high signal intensity for each sugar in this analysis because all different signals from one sugar come together to form one strong signal. And another advantage of this analysis is application to isotope composition analysis of each sugars. A previous meteorite investigation found a very important characteristic of the sugar-related compounds. These are sugar-related compounds, sugar acids and sugar alcohols. And that is the important characteristic is the enrichment of carbon-13 in these molecules. Terrestrial biological sugars and most other organic compounds, terrestrial organic compounds, are depleted in carbon-13. While meteorite amino acids are known to be enriched in carbon-13, and this previous work found that the sugar-related compounds are also enriched in carbon-13. So this characteristic is very useful indicator to evaluate that the detected molecule, detected sugar-related compounds in sugar is extraterrestrial origin or biological contamination. We analyzed 3 meteorites, these 3, NW801, NW702R, and the Murchison meteorite. Just inner part of the meteorite were used, and the mild extraction by hydrochloric acid in water under ambient temperature was conducted. The extracts were derivatized as explained and analyzed by gas chromatography, mass spectrometry for sugar identification, and also conducted the compound specific carbon isotope analysis by gas chromatography, combustion, isotope ratio, mass spectrometry. And we also analyzed the organic residue for the evaluation of maturation of organic compounds in these meteorites, and also analyzed minerals to investigate the extent of aqueous processes in parent asteroid of these meteorites. So this is the result. This is a result. We detected ribose, licosos, arabinose, and xylose. And from Murchison, from the Murchison meteorite and NWA801 meteorite. These sugars were not detected from NWA 702 meteorite. We also conducted laboratory sugar synthesis by formaldehyde reaction. An aqueous solution containing formaldehyde, glycolaldehyde, ammonia, calcium hydroxide was incubated at 90 degrees C for fixed durations. And the final formation of ribose, lixisos, laminose, xylose, these pentoses. When we compare the yield of the product sugar, lixisos and laminose formed more abundantly than ribose and xylose and arabinose formed more abundantly than ribose and lixisos. And this characteristic is similar to these two meteorites. And suggesting this similarity may suggest the formation of these molecules, these meteorite sugars, by the formaldehyde reaction in the parent body asteroid or former steps in interplanetary environments. We analyzed carbon isotope compositions of sugars from the two meteorites and a soil sample from Murchison, Australia. Sugars in Murchison soil and sugars in terrestrial biological samples all have negative carbon isotope compositions. This means that these sugars are depleted in carbon-13. Conversely, sugars detected from meteorites are enriched in carbon-13. This characteristic is similar to meteorite amino acids and the meteorite sugar acids and sugar alcohol. So the carbon isotope composition really deny the possibility of the terrestrial contamination. When we compare the components of sugars in the three meteorites, we cannot find clear relations to asteroidal aqueous processes and the type of meteorites. For example, these two meteorites are CR chondrites, the same group, but have completely different contents of sugars. And these two meteorites have similar aqueous processes, extent of aqueous processes in asteroid, but have different complete contents of meteorite, contents of sugars. So it's not clear so far. Now we are analyzing more meteorites and also analyzing chirality of these sugars. A previous study reported an interesting chiral enrichment, chiral enriched D-sugar acids. We are analyzing actual sugars, whether this is also true for sugars. So in conclusion, meteorites delivered extraterrestrial sugars, including ribose, to the Earth, and most likely to prebiotic Earth. But we need further investigation to estimate the amount of sugars from space and the amount of sugar formed on prebiotic Earth for more comprehensive understanding of ribose accumulation on prebiotic Earth. Well, I'm stopped talking. Thank you, thank you all audience tonight.
Thank you very much, Yoshihiro. So now we are at the end of our talks for this session, and I suggest we have one or two questions to Yoshihiro right now, and then we can discuss also questions to, or ask also questions to the other speakers, or any questions you have now, please feel free to unmute yourself and ask a question.
Yoshihiro, I'm very much interested in your sugar analysis. So what about peptides, proteins?
Well, I didn't do the analysis of peptides.
But I mean, now with experience from the sugars, what are the prospects?
Peptides? Yeah, so the amino acids, many amino acids have been detected from meteorites. And I think so, and also asteroid condition is originally water rich, and later the water disappears. I think some very small part of amino acid might be from peptide. This is just a guess.
Okay.
Uli,
Uli,
maybe
you're
it's
muted.
Sorry. Yeah, feel free to ask a question now. I see Ramkrishnamurti, you have unmuted yourself. Did you want to ask a question? You can raise your hand and then you automatically go to the front and it's easy to see you. Sorry. Of course, okay. The other possibility is that now after the session, you can go out of the auditorium and you can, oh, okay. We have questions from Ricardo Mauricio Cabrera.
Can you hear me?
Yes.
Thanks for your great talk, Yoshihiro. My question is about the foremost reaction, because it tends to form complex sugars, even with some ramification, with some branching. Have you found any of these complex molecules, sugars?
Well, from meteorites, we didn't see such complex branched sugars. And we also investigated the sugars, but we didn't find the branched sugars. The branched sugar can form in the usual thermostat reaction, but yeah, we didn't see any meteorites.
Thank you.
Thank you.
This is Ram Krishnamurthy. I have a question. Can people hear me?
Yep.
Yes.
Yeah. So this is general for for all the speakers, not just for this one, but in the spirit of the carefulness of the first speaker, who showed interpreting the early Earth rock record in terms of early life, was very careful in saying what are the pitfalls. Now here we are identifying molecules which apparently have a connection to life on Earth, but the amino acids or the sugars that are formed in such small amounts among millions of compounds, if you will, to try and make a direct connection between that and origins of life may be a stretch. And I just want to put it out there. I'm not disputing one way or the other. It's just that trying to make these connections directly to the type of molecules that are found on life today or found in life today, one has to be a little careful. I just want to leave that thought out there. That's all. Thanks.
Thank you, Ram. Dieter Braun has a question.
Yes, just a short question to figure out how quantitative, if these sugars are intermingled with geological matrices, other materials, can you become? Are you confident that you can quantify all those steps in between, or where comes the most error bar in your workup of the molecules?
Even so, the estimated amount of sugars on prevaricators?
Yes, I think that's the question.
Okay, so this is just a simple estimate here. For example, we support the late heavy bombardment